Today we are going to have a lecture about the hemolysis, the breakdown of RBCs. You know, in everyone, normally red blood cells circulate in the blood about 120 days, right? 120 days. After 120 days, red blood cells are destroyed within our body. Is that right? And their breakdown products are handled by our body. So for making a discussion about the red blood cell destruction, we have made this diagram. Here is your circulatory system. Here is the bone marrow house, from bone marrow from where RBCs are produced and coming to the general circulation, right? And spleen, I have shown it here, in which RBCs are destroyed. Here is one nephron, which represents the renal function or kidney function. Here we have liver and GIT. So let's start from the very beginning, that red blood cells in adult are produced by the bone marrow. All of you know in the bone marrow there are stem cells, and those stem cells break down into myeloid stem cells and lymphoid stem cells. Then one of the myeloid stem cell lineage go into erythroid line, right? And erythroid cells, right? Let's suppose put it here that this is bone marrow stem cell. Stem cell divide into, yes, lymphoid stem cell and myeloid stem cell. Myeloid stem cell and lymphoid. Myeloid stem cells go into multiple lines. These cells multiply and they break down into, yes, there are three types of cells. Who will tell me? Myeloid stem cell, they break down into granulocyte monocyte series, erythroid and megakaryocyte series, and yes, basophil series, right? Out of this erythroid megakaryoid cells, they divide into two series, erythroid series and megakaryoid series. Erythroid series cells, right? These cells of erythroid series, they basically proerythroblast, then they go into normoblast, early, intermediate, late, reticulocyte, yes, reticulocyte, and eventually what comes out? Red blood cells. Is that right? So this is a process, hematopoiesis process, which is going on in the bone marrow. Is that right? Now, this is erythroid series. Red blood cells are constantly being produced by the bone marrow in adult. When red blood cells come into general circulation, right, they keep on circulating for about 120 days approximately. Right? During that, they are doing their major functions. The most important function is that they are carrying hemoglobin and transporting oxygen from the lungs to the peripheral tissues. Is that right? Now, these red blood cells, after approximately 120 days, they are destroyed in our body. Why they are destroyed? Why the red blood cells are destroyed after 120 days? The reason being, red blood cells do not have nucleus. During the erythropoietic process, red blood cell loses nucleus, right? And because red blood cells do not have the nucleus, right, they are unable to synthesize new proteins. Because if you have a nucleus, then you have DNA. And if you have DNA, then you can make RNA. And if you can make RNA, then you can make proteins. But because red blood cells do not have nucleus, they do not have DNA, and they do not have any RNA, so they cannot make new proteins. So whatever they are packed with, whatever proteins they are packed with, they keep on circulating. Now, approximately 120 days, many of their proteins get denatured. Their proteins become denatured, and they don't function well, right? Let me show exactly what happens. In the red blood cell, if I'm making one red blood cell large, right, in the inner side of the red blood cell, there are special type of proteins, which I will teach you in detail later. And these proteins, there are proteins which are in the membrane of the red blood cell, and these proteins are interlinked with internal proteins. And all these proteins are cross-linked with each other. This network of proteins, structural proteins, which are holding the membrane and making a network inside, these are called cytoskeleton of red blood cell. What are these proteins called? Cytoskeleton of red blood cell. Most important protein among them are spectrin, right? And here in the membrane, ankyrin. So spectrin and ankyrin and many other proteins make a cytoskeleton of red blood cells. Is that right? Now what happens? What is the function of this cytoskeleton? Cytoskeleton hold the membrane of red blood cell with the substance of red blood cell. Of course, in the red blood cell, what is here? There's a lot of hemoglobin and other cytoplasmic material. Red blood cell has extra membrane. If you look to the red blood cell from the sides, right? What is happening in the red blood cell? It has less cytoplasm and more cell membrane. Due to this reason, due to extra cell membrane, it is having biconcave shape. What it is having? Biconcave shape. Is that clear to everyone?
Yes.
Now, why nature has provided extra membrane to red blood cell? So that it can undergo gas exchange. It has more surface for oxygen absorption and release. So it is beneficial. But it is an example of a small woman with a very large dress. Have you seen there are some dress which women love to wear at their marriage parties? The woman may be small, but dress may be very, very large. Red blood cells are like that. That they are having less substance and more bigger dress. Is that right? Now what happens? When RBCs red blood cells are circulating into circulation, listen now carefully. When they are passing through circulation, some points in circulation are very narrow. For example, some capillaries are very narrow. Especially some circulatory point within the spleen are very, very narrow. When RBC have to pass through those narrow points of spleen, they have to twist and turn like acrobatics. Is that right? And they have to do the acrobatics and pass through those narrow tunnels. Am I clear? Now, RBCs have extra membrane. They have a lot of extra membrane, so they can twist through those areas. And these proteins are very, very flexible. These cytoskeletal proteins are very, very flexible. Due to their flexibility, RBC membrane and RBC cell can change its shape. Now you imagine an old RBC, which has been circulating for 120 days. It has been insulted by different chemical substances in the blood. Its proteins has become denatured. If proteins of this cytoskeleton are denatured, do you think they will be as flexible as they were in fresh? No. They are like old man bones and old man joints, not like a young man. So what happens when RBCs keep on circulating for a long time, when these proteins get denatured, normally in the cells, when proteins are denatured, nucleus orders new protein and catabolizes old protein. But because here is no nucleus, they cannot replace the denatured protein with new proteins. So the problem is that these denatured cytoskeleton become stiff, hard, less flexible. Now when RBCs will pass through narrow points, can they twist and turn through that?
No.
So what happens? They get stuck at multiple places. Sometimes they are, as I told you, originally they are what? Small substance with a big dress. When they get stuck with some membrane, they try to go through that, they leave a piece of membrane there and pass away. So when RBCs become older, their proteins, cytoskeletal proteins are denatured, they cannot hold the membrane properly, they cannot keep the cell properly flexible. So whenever in narrow point RBCs get stuck, sometimes it leaves a piece of membrane there and run away. As the woman with very light dress, if her dress is caught into something, she just go away and leave a piece of dress there. So when repeatedly membranes are a little bit lost, do you think it will remain biconcave? No. A piece of membrane gone, remaining membrane fuse. So as time will pass by, as membrane is being lost, it will become globular. It will become spherical. Is that right? So this RBC, which was originally like this, with membrane loss, it will become like this. Because the membrane is less, but inside the substance is the same. So it becomes spherical. When it will become spherical, it will become like an obese man, fat man. Can it now twist and turn? No. So eventually it gets stuck to some narrow point. It's stuck there. Now it cannot twist and turn. This is one problem with old RBCs. Second problem with old RBCs is that in RBCs membrane, there are special pumps which keep on throwing the sodium out. As RBCs get older, these sodium handling channels and pump proteins also become denatured. So they cannot get the sodium out. So little sodium is accumulated in, and sodium hold with it water. So RBCs swell up. Now you imagine those very young, fresh RBCs with extra membrane, highly flexible proteins, twisting and turning around with acrobatics and going through narrow capillaries. When they become old, their membranes are less. Membrane surface is less. Their proteins are hardened. And water is accumulating in because sodium is accumulating in. So they will swell up. So what happens? These become more flexible red blood cell or less flexible red blood cell? Less flexible. So what happens that as RBCs become old, they become less flexible red blood cells. Now in the spleen especially, when blood enters into spleen, suppose this is arterial blood, and here it is going into spleen, this is venous blood which is going out. I'm making a very small diagram. Arterial blood goes into arterioles and smaller. Eventually very small channels come. These very small channels are called, blood is going into those channels. These channels are called cords of Billroth. So in the spleen, there are very narrow vascular channels which are called cords of Billroth. Now, actually what happens, if I make one cord of Billroth here, it has endothelial cells here. What is this? Endothelial cells. Okay, I will make a different color. This is the endothelial lining of cords of Billroth. Endothelial cells here. Right? Normally what happens, physiologically, RBCs will pass through these points. They squeeze through this point outward into splenic sinusoids. So what happened, through the walls of cords of Billroth, through very special, this is one long endothelial cell, and this is another long endothelial cell. In between the endothelial cell, there are small slits. What are these? Long slits. So what happened, this is a cord of Billroth, and their endothelial cells and longitudinal endothelial cells are having slits. RBCs have to go through these slits out into sinusoids and then into veins. Now RBC which is very large, or which is not flexible, or which is stiff, which is old, do you think that can pass through the slit? No. So old RBCs, these are, you can say, special operators to check the RBC's flexibility, biological operators. So old RBCs get stuck into that. Now, when old RBC is trying to go from here and stuck here for a long time, there are special type of RBC eater here. What is this? Macrophage. When RBC remain there stuck for a long time, they will just grab the RBC and eat. In this way, from circulation, old RBCs are constantly being removed and new RBCs are constantly being produced. Is that right? This is normal mechanism that in the spleen, right, in the cord of Billroth, older RBCs get stuck. And then in the wall of the cord of Billroth, there are macrophages. When RBCs are stuck there longer, macrophage will phagocytose those red blood cells. And if there's fresh red blood cell, it will try to eat, but that will squeeze through and run away. Am I clear? Now, once RBC is eaten up by macrophage, this is the macrophage, then within the macrophage, what happened to red blood cell? Now we have to talk about what happens to red blood cell within the, macrophage. So this is your spleen, and in the spleen, I'm going to draw one macrophage. Right? What is this? This is one macrophage. I'm showing one macrophage in the spleen now, right? And we'll see when red blood cell is taken by the macrophage, what really happens to this red blood cell, right? Here is red blood cell. Naturally, within the macrophage, red blood cell will be destroyed. Now, when it is destroyed, what substances it will release? It will release its membrane, and it has cytoplasm. What other component is there? Cytoplasm. Is that right? Membranes are made of lipids and proteins. Proteins will be broken down into amino acids, and lipids will be broken down into fatty acids. and they will be reutilized. Either they will be released into circulation, amino acids and fatty acids, and they can be reutilized to synthesize more new proteins or to fats. Clear? Then with the cytoplasm, we are again left with proteins, especially enzymes. In the cytoplasm, there are enzymes, and these enzymes will also break down into amino acid, and they will be also used. But special protein which is present in red blood cell, we should talk about that. The very special protein which is present in red blood cell is hemoglobin. So when daily RBCs are being destroyed, it means actually hemoglobin is constantly being destroyed. When we say daily hematopoiesis is occurring and daily RBCs are released, it means RBCs packed with hemoglobin are released, right? And when RBCs are destroyed, we say RBCs along with their hemoglobin content are destroyed. Remember, hemoglobin is highly toxic molecule. Hemoglobin is a highly toxic molecule. That is why hemoglobin is kept within RBCs. It is in the cover of RBCs. Hemoglobin does not circulate normally free in the blood. Even though it's a very wonderful protein, it transports oxygen, but it is highly toxic. So it is kept within the membranes of RBCs. And here when RBCs break down within the macrophage, so hemoglobin is not released into blood, it is released within the macrophage. Right? Here we'll safely handle with the hemoglobin. So let me tell you that here is your hemoglobin. Hemoglobin has, what is the basic structure of hemoglobin? You must be knowing that hemoglobin has, I will make its basic structure here. It has this is one pyrrole ring, and how many pyrrole ring? 1, 2, 3 and 4. 4 pyrrole ring together are called protoporphyrin. I mean, this is one pyrrole ring, this is other pyrrole ring, another pyrrole ring, another pyrrole ring. When these 4 pyrrole rings are together, this is called protoporphyrin. And in the protoporphyrin, in the center of the protoporphyrin, here we have what thing? Iron. What is there? Iron. So protoporphyrin plus iron is called heme. Protoporphyrin plus iron is called heme. And with the heme, there is a globin chain. Here, what is it? Globin chain, which is made of amino acids. So this is the basic structure of hemoglobin monomer. So hemoglobin has heme and globin. Now, first of all, within the red blood cell, hemoglobin breaks down into 2 components. It breaks down into globin and it breaks down into heme. Now, this is the heme component and that is the globin. Globin is broken down. This globin is broken down into amino acids. It is broken down into amino acids and they can be utilized. Now we are left with the last thing to deal with. What is that? Heme. Heme is what? Heme is one protoporphyrin ring with iron. So heme will be divided into 2 components. That is iron. Iron will be released out of heme. And what is left behind? Protoporphyrin ring. That ring is opened. When that ring is opened, it is like this. Are you understanding? We were destroying the RBC and we destroyed the membrane, we destroyed cytoplasmic enzymes, we were left with the hemoglobin. Hemoglobin was broken into heme and globin into amino acid. This heme component was open, iron was removed, we were left with the open protoporphyrin ring. Now this iron molecule is also toxic molecule. It cannot be kept free into the cell. So this iron molecule, as soon as it is produced, suppose this is the iron molecule. Immediately, suppose here is the iron molecule, which is brought out of it. Here was iron originally. When it is brought out, macrophages produce another protein, and that protein bind with this iron molecule. So this iron molecule with that protein is equal to that protein plus iron molecule. They make a complex. This complex is not toxic, but free iron is toxic. This protein is called apoferritin. So apoferritin plus iron is equal to this combination, and this combination is called ferritin. What is it called? Ferritin. And if there's too much ferritin, many ferritin molecules, they fuse with each other and they make polymers. What they are making? Polymers. Many ferritin molecules fuse together and they make polymers, and this polymer of, what was this? Iron with apoferritin, and now they are making polymer. Whole this molecule is called, yes, what is this? Very good. This whole molecule is called hemosiderin. So we are talking about the normal breakdown of RBCs. So RBCs, heme breakdown into iron and other compound. Iron bind with apoferritin, make ferritin. Many ferritin molecule fuse together and make hemosiderin. No problem? In this way, iron can be stored here, and later on it can be released to and carried to the bone marrow to make new hemoglobin. Is that right? Now we go back, we talk about this compound. This compound is called biliverdin. What is this compound called? Biliverdin. Right? This biliverdin, by action of another enzyme, right, it converts into a compound which is called bilirubin. What is this called? Bilirubin. Now this is our bilirubin. So bilirubin is one of the breakdown product of hemoglobin. This is bilirubin. Now this bilirubin is again highly toxic molecule, and we should dispose it out of the body, even though it is produced within the macrophages by the breakdown of red blood cell. But this is a highly toxic molecule, and it should be safely thrown out of the body. Right. Now this bilirubin, as soon as, okay, I will make bilirubin now onward by this color. This is equal to this. This is bilirubin. This bilirubin will come into general circulation. What is it? Bilirubin. Now this bilirubin molecule, when macrophage eat the RBCs, they release bilirubin. And where RBCs are destroyed, most of them are destroyed in spleen, but some of them are destroyed by the macrophages of liver, and some of them are destroyed by the macrophages which are present in bone marrow. Right? And because this breakdown of red blood cell is within the circulation or outside the circulation? Outside the circulation. This normal breakdown of red blood cell is not into circulation, it is in the macrophages. So it is outside the vessels. That is why this type of normal breakdown of RBCs is called extravascular breakdown of RBCs. What is this? Extravascular breakdown of RBCs. Now, when macrophages have broken the RBCs, they release bilirubin as a waste product. This will come into blood. As I told you, bilirubin is a highly toxic molecule, right? And it should be carried to the liver to be destroyed. It should be carried to the liver to be eliminated out of body, right? But bilirubin is a very small molecule. It has a tendency to leak from capillary wall into tissues. Bilirubin is a very small molecule, and it has a tendency to leak from capillaries out. So what happens? Liver produces a special type of bilirubin transporters, and bilirubin transporters are normally produced by hepatocytes and released into general circulation. What is this? This is a special protein produced by the liver, and it is normally present in your blood. As soon as bilirubin comes out, this protein which is coming from the liver will bind the bilirubin. What is this protein called? Of course, this is simply called bilirubin binding protein. So liver produces bilirubin binding proteins. This is albumin and pre-albumin type. And what really happens, that bilirubin bind with the, as soon as it comes into circulation, it will bind with bilirubin binding proteins. Now it is a bilirubin protein complex. Now this bilirubin is called, this bilirubin which is now complex with protein, what is the advantage of it? Advantage of complexing the, binding the bilirubin with bilirubin binding protein is, this complex is very large. And this does not easily leak out into tissues. It is now trapped into circulatory system. And whenever blood will pass through liver circulation, right, in the liver cells, here there are special type of receptors expressed. These are the receptors which are expressed on the surface of hepatocyte, right? And these receptors are facing towards the blood. Actually, they love to bind bilirubin, binding protein and bilirubin. So what happens, the bilirubin binding protein bind with the bilirubin, right? And this complex cannot leak out of circulation, and even it cannot go out into urine because it cannot filter out through that. I should make a diagram here. This is bilirubin binding protein, and here is your friend, yes, bilirubin, and actually they cannot filter out. So this form of bilirubin cannot appear into urine, right? And this bilirubin along with its protein will eventually pass through this area, and here it will be caught. Here it will be caught, and bilirubin binding protein will bind over here, right, with this receptor, and of course bilirubin will stick over here, and then this is internalized. In this way, bilirubin is transported to the hepatocyte, to the liver cell. This bilirubin, which was binding with the proteins, it is conjugated bilirubin or unconjugated bilirubin? Who will tell me? Unconjugated. It is unconjugated bilirubin. What do you think? It is conjugated? Unconjugated. It is unconjugated, yes. This point you have to remember. This is unconjugated bilirubin. Complexing of bilirubin with plasma protein is not called conjugation. Conjugation is a process in which bilirubin bind with glucuronic acid in the liver cells. So this is called unconjugated bilirubin, and this unconjugated bilirubin cannot leak into urine because it's a very large complex. Is that right? Now, once unconjugated bilirubin is taken into hepatocyte, in the hepatocyte there is a endoplasmic reticulum. You know endoplasmic reticulum? Endoplasmic reticulum. This bilirubin is transported to the endoplasmic reticulum. In the endoplasmic reticulum, there is a very special type of enzyme here, and this enzyme, this is called glucuronide transferase. What is this enzyme called? Glucuronide transferase. This enzyme holds one glucuronyl group and bind it, transfer it to the bilirubin. So when bilirubin is passing through this area, endoplasmic reticulum, and it comes out of it, now this bilirubin is loaded with, yes, this bilirubin is loaded with what? Glucuronic acid, right? So let's make glucuronic acid this color, glucuronic acid. Now this bilirubin is conjugated with glucuronic acid. Now it is called conjugated bilirubin. Now this conjugated bilirubin is highly polar. It is a highly charged molecule. It cannot dissolve into lipid membrane. So it cannot diffuse out of hepatocyte. It cannot diffuse out of hepatocyte. So it is actively transported by special active transporters. There are very special active transporter present in the membrane of hepatocytes. So this special active transporter transport this conjugated bilirubin to the, what is this? Biliary duct system, biliary canaliculi. So eventually we find our bilirubin which is conjugated and which has reached to the biliary canaliculi. Now it is conjugated bilirubin. This conjugated bilirubin passes through this area to the common bile duct and right and left hepatic duct, then common hepatic duct, then common bile duct, and eventually, what is this? This is pancreatic duct and this is common bile duct. They make together a duct which opens into duodenum, second part of duodenum. So this bilirubin will come into second part of duodenum. And when this bilirubin reaches here, this conjugated bilirubin within the GIT will be acted upon by different enzymes and bacteria. When it is acted upon by different enzymes and bacteria, it becomes modified. This modified bilirubin is called urobilinogen. This is acted upon by, conjugated bilirubin is acted upon by bacteria and enzymes in the GIT, and it is chained, right? It is called urobilinogen. Some people call it stercobilinogen. Actually, urobilinogen and stercobilinogen are same molecule. What really happens, most of it will pass through the fecal matter out. You know where it goes. This component of altered conjugated bilirubin, either you call it urobilinogen here or stercobilinogen, but what component of it appears into fecal matter, that is called stercobilin. Stercobilin. So what is stercobilin? It is basically modified conjugated bilirubin. Is that right? No problem here. This is responsible, stercobilin is responsible to give yellow-brown color to your fecal matter. Is that right? Now, most of it, this urobilinogen or stercobilinogen goes into fecal matter. So, most of this bilirubin molecule is eliminated, goes out of the body through the fecal matter. But a very small part of urobilinogen is absorbed from here. This is your portal circulation. What is it? Portal circulation. This is liver circulation, okay? Now what really happens, that very small part of urobilinogen is absorbed from here, from the GIT, and it goes through portal circulation into liver. When it is passing through the liver, most of this urobilinogen is taken up by liver cells again, and liver cell again pump that into bile system, and it will again go down. A very small part of urobilinogen may go into general circulation. This urobilinogen may pass through here, and it can cross the glomerular filtration system, and this will eventually appear into urine. So normally your urine has a very small amount of urobilinogen. Am I clear? There's no problem here, right? Now I will explain that if there is excessive breakdown of red blood cells, what will happen? Normally, when red blood cells are normally broken down in your body, small amount of bilirubin which is coming into circulation, it gets bound with the plasma proteins, carried to the liver, and the liver it gets conjugated. Conjugated bilirubin goes to GIT. Most of it goes out as stercobilinogen in fecal matter. Some of it is reabsorbed as urobilin, but while it is passing through the liver, it is again pushed into biliary drainage system. Very small percentage escape into general circulation. From there, traces may come into urine normally. This is the normal breakdown of red blood cells. Now let's have a break and then we'll continue.